Yeah, hi and hello everyone. I'm Partha from the Biomix team. Would like to welcome you all for today's talk. Today we have another great personality in our platform, an eminent scientist from Indian Institute of Science, which is a leading topmost institute in our nation, and first in NIRF, which is also an institute of eminence, from where we today have Professor Dr. Kavita Babu with us for today's talk. On the Biomix team, I would like to extend my warm welcome to Dr. Kavita Babu. She is here today with us for her talk on drinking and dopamine. What's the connection? She is an ex neuroscience, so she will be newer areas in neurosciences. At this moment, I would like to thank Dr. Kavita Babu for accepting my invitation. It's really a pleasure for being your presence in our virtual platform. And welcome all the participants who joined with us for today's talk. We used to have question and answer session at the end of our presentation. You are most welcome to place your questions in the chat box of the YouTube. I and a team are here to bring your queries to our speaker. And you can place your feedbacks as comments in the same chat box of the YouTube. Prior notifications for the upcoming webinar talks will be updated in our Telegram channel. You can get access through the Telegram channel as well. I thank all our subscribers from various fields of biological sciences for their active participations and made these consecutive talks in a successful. And I am really amazed that we have surpassed 3,000 subscribers in YouTube channel for our talks. This credit goes to our broad audience, right from undergraduates, postgraduates, research scholars, postdoctoral community, and all other faculties of life sciences stream who made this event a great successful. Now, on starting our session, I am very much delighted to introduce our today's speaker, Dr. Kavita Babu. Dr. Kavita Babu is currently working as an associate professor and Wellcome Trust DBT India Alliance senior fellow at Center for Neurosciences in Indian Institute of Science, Bangalore, India. She is also a lien from Indian Institute of Science Education and Research, IISER, Mohali. She was a postdoctoral fellow at the Department of Molecular Biology in Massachusetts General Hospital, USA. She obtained her doctorate in developmental and cell biology from National University of Singapore. Dr. Kavita Babu's laboratory is currently studying the signaling role of cell adhesion molecules at neuromuscular synapses in the free-living nematodes, Sinorhabditis elegans. Her laboratory is also interested in understanding how neurons communicate with each other using neuropeptides and how these neuropeptides-based circuits allow Sinorhabditis elegans to modulate for locomotion under different conditions. She is a recipient of several awards such as a B.K. Bachawat Travel Award in 2020, Wellcome Trust DBT India Alliance Senior Fellowship Grant in 2020, DBT-JRF National Women Scientist Award Young category in 2020, Wellcome Trust DBT India Alliance Intermediate Fellowship Grant in 2012, and DBT Innovative Young Biotechnologist Award 2012. With this brief intro, now I would like to hand over the session to Dr. Kavita Babu for her talk on drinking and dopamine. What's the connection? Ma'am, over to you.
Hi, you can hear me, I'm guessing, right?
Yes, ma'am.
Yeah, okay, fantastic. So first I would like to thank the Biomix team and especially Partha for the generous introduction. And I'm going to tell you a little bit about the work that we do in our lab. I'm going to go from here and I'm going to go full screen to my presentation towards the end of it.
Yeah. Yes, ma'am. It's all clear.
All right. Human brain is required for all our activities. Walking, talking, everything requires functioning of our brain. And the brain is made up of large neurons that are called neurons that form connections with each other. These connections between neurons are called synapses. These neurons actually talk to each other through synapses. We know that you have an electrical signal that comes along the axon of a neuron, causes of from the neurotransmitters to receptors that are present on the postsynaptic cells, then another neuron or the muscle cell to which the neuron forms connection. This then causes changes in the activity of the postsynaptic cell, and this process is innumerable times, and this is what allows us to do everything that we do throughout the day for years and years. And this process of neural communication is conserved across all systems that have neurons and all multicellular organisms that can do multiple processes. In the laboratory, we use a small free-living nematode, which is called Tenorhabapis elegans. And we use this organism to study aspects of how neurons actually communicate with each other or talk to each other. So why Tenorhabapis elegans? What is known is that the human brain has more than 100 billion neurons. You compare that to C. elegans, which has around 300, which has actually exactly 302 neurons. The human brain has more than 10 trillion synapses. C. elegans has around 7,000 synapses. However, when you look at the number of genes, humans have around 25,000 genes, and C. elegans has a very close number of 20,000 genes. And a lot of the genes, be it for transcription factors, be it for enzymes that are required to make neurotransmitters, proteins that encode for neurotransmitter receptors, cell adhesion molecules, a lot of these molecules are conserved between C. elegans and humans, making it a really interesting system what these molecules are doing. So I just wanted to give a brief idea as to what C. elegans looks like. So this is a microscope under which we look at the C. elegans, and this is how C. elegans looks like. So this, I don't know, see my arrow. This is an older C. elegans. These are younger C. elegans. This is an adult C. elegans. You can see here the stars indicate C. elegans eggs. These are really young C. elegans in the early larval stage. And just to give you an idea as to how the life cycle of C. elegans progresses, you essentially have embryogenesis followed by L1 to L4, which is then followed by the adult stage. The adult then lays multiple around 300 to 400 eggs and gives rise to more C. elegans. The C. elegans that occur in the wild are largely hermaphrodites and form clonal populations that are identical to the mother. But you can also make male C. elegans and mate the male C. elegans with the hermaphrodite. Good system to study a lot of neurogenesis. So this is C. elegans expressing DF3, and this is a live worm, and you can see that you can actually see DF3 neurons and in axons, and you can image the system quite easily because it's a transparent worm. So what do we study in the lab? As Parth had alluded to, a large part of our lab is looking at how cell adhesion molecules affect signaling at the neuromuscular junction. Looking at is how motor neurons that synapse onto muscle actually allow for communication between neurons and muscles, and who are the molecular players. However, in today's talk, I'm going to be talking about neuronal circuits that modulate locomotion. So this is another area that some of my lab members are studying. They're interested in understanding essentially how neuropeptides modulate locomotion in C. elegans. So today's talk is largely going to be about how dopamine changes in dopamine makes the worm more susceptible to F. What is dopamine? Dopamine is a biogenic amino neurotransmitter. Dopamine regulates many important physiological functions like emotion, coordination of locomotion, cognition, and a lot of other functions. Easy to study, or it's very easy to study dopamine in dopaminergic neurons in C. elegans is because there are only 8 dopaminergic neurons in C. elegans. compared to the many, many neurons that are present in the human. And these 8 can be seen very nicely here. You have 4 CET neurons, 2 ADE neurons, which are in the head of the animal, and that is 2 PDE neurons, or one set of PDE neurons that is more posterior in the animal. So how does dopamine actually function as a neurotransmitter? You have tyrosine that is converted to dopa and dopamine, which is put into vesicles. This is then released from the presynaptic neuron, and this release is affected by the dopamine transporters and dopamine synthesizing enzymes like DAT1 and CAT1. Apart from this, you also have dopamine receptors that sit on the presynaptic neuron like DOP2, and these are autoreceptors that regulate dopamine levels from the presynaptic neuron. Once dopamine is released from the presynaptic neuron, it binds to postsynaptic receptors like DOP1 and DOP3 and allows for activation of the postsynaptic neuron or postsynaptic cell. A lot of work has also shown that alcohol affects neurotransmitters. Some of you might have seen in life or in movies, when a person has too much alcohol, they start stumbling. So it's essentially alcohol is affecting the nervous system in a way where coordination is affected, or coordination is not like in normal scenarios. And a lot of work has shown that alcohol affects the glutamate neurotransmission, GABA neurotransmission, and dopaminergic neurotransmission. And many of these pathways actually merge into each other, and alcohol affecting one could actually affect another pathway at the same time. So what is known about the relationship between alcohol and dopamine? A lot of work has been done before, and a lot of work is also quite contradictory. And I'm going to show you a little bit of that in this slide. So if one were to look at the brain of alcoholics, one sees that there is a decrease in some of the dopamine pathway molecules, like the dopamine receptor, D1 receptor, or the dopamine transporter, DAT transporter. Work has also shown that in the presence of ethanol, more dopamine could be released from the dopaminergic neuron, indicating that dopamine might be affecting alcohol might be affecting signaling in the dopaminergic neuron in multiple different ways, depending on the system that you're looking at, the organism you're looking at, the part of the brain you're looking at. So one has to be very careful when one is actually studying these aspects of how alcohol might affect neurotransmitters. So what we wanted to look at was, we wanted to look and see whether there was any link between dopamine and alcohol in CLS. In order to do that, we used a protocol that has been previously published in 2003, where we first looked to see what happens when you add alcohol, you treat wild-type C. elegans with alcohol. So what we see is that when you take the worms, take them from plates that have food to plates without food, which has alcohol, what you immediately see is that within 10 to 12 minutes, all the worms would have parried. After around 30 to 40 minutes, they start recovering. And by the end of 1 hour, the normal wild-type C. elegans worms recover all completely, and they are completely indistinguishable from the animals that are on plates without alcohol. So what we wanted to do was, we decided to look at how quickly the worm paralyzes and recovers in mutants that lack different molecules in the dopaminergic pathway. We did the screen for multiple dopaminergic molecules, and we only found one mutant in DOP2 that showed a very significant phenotype in the presence of alcohol. So this is wild-type animals which have been treated with alcohol for 2 hours, and you'll see that they move very normally. This movement is exactly what you see in wild-type animals without alcohol treatment. However, when you look at mutants in DOP2, which had been treated with alcohol, we saw that they're finding it difficult to move. Anterior region of the body is trying to push the posterior region forward, and they keep moving around in circles because they're completely unable to move normally, even after 2 hours of alcohol treatment. So when we quantitated this phenotype, we decided to use two ways of quantitating the phenotype. We looked at the number of body bends from the anterior region of the worm and the posterior region of the worm. And you can see that the number of body bends in the posterior region of the worm is very, very few when compared to wild-type animals, which were also treated with alcohol. While the number of anterior body bends were also less in comparison to the control, but not as badly affected as the posterior body bends. We also looked at the amplitude of the body bend, which is essentially the distance between a line that is drawn through the body bend and the highest point away from it. And again, we found that both anterior and posterior amplitude body bends were defective. However, the posterior was far more defective when compared to control animals, while the anterior did not have that much. Okay, so we see this phenotype in the presence of ethanol. What happens when we were to just look at wild-type animals with and without ethanol? And when we did this experiment, we found there was no significant effect after 2 hours of ethanol treatment for wild-type animals. So you can see compared to wild-type animals without ethanol, wild-type animals with ethanol, 2 hours of ethanol treatment show no defects either in the number of body bends or in the amplitude. The next question we asked is, when we look at DOP2 mutants, the phenotype in the presence of ethanol, do we also see a phenotype in the absence of ethanol? So we tested wild-type animals without ethanol with DOP2 mutants, also without ethanol. And we found that there was no difference. They looked just like wild-type animals. In the absence of ethanol, they move normally, the amplitude of body bends, the number of body bends, everything was very similar to what we see in the control of wild-type. The next question we decided to ask was, if this is actually because of defects in DOP2, can we put back DOP2 into these animals and rescue the phenotype? And we found that yes, that was indeed the case. When we put back the DOP2 transgene into the DOP2 mutant and looked at animals treated with ethanol, we found that they reverted back to looking just like wild-type animals. So this is wild-type animals, DOP2 mutant animals in the presence of ethanol. This is putting back DOP2 mutant, DOP2 into DOP2 mutants. You can see they behave just like wild-type animals. They're indistinguishable from wild-type animals, both in the number and the amplitude of wild-type. So far, what I've shown you is that you have defects in animals that lack DOP2. And what is DOP2? DOP2 is essentially an autoreceptor that sits on the dopamine neurons and is thought to regulate dopamine. So in the presence of ethanol, we find that when you lose DOP2, the worm is essentially is unable to move normally in the presence of ethanol and is moving around in circles, dragging it. So the next question we decided to ask was, does DOP2 actually show this phenotype because it is modulating the levels of dopamine? So in order to answer this question, we decided to see what happens to the DOP2 mutants when we remove dopamine completely. There's a mutant called CAT2, which is required to convert tyrosine into L-DOPA. So CAT2 is a tyrosine hydroxylase that allows for conversion of tyrosine to L-DOPA. The L-DOPA is then converted to dopamine by another enzyme. So you can imagine now, if you don't have CAT2, there will be more tyrosine and less L-DOPA. So first we decided to look at CAT2 mutants. So if CAT2 mutants showed a similar phenotype like the DOP2 mutant, we would say that maybe what is happening in DOP2 mutants is that there's less dopamine. Okay? But what we found was that when we looked at CAT2 mutants, they looked just like wild type. They were able to move without any problem after 2 hours of ethanol treatment. And both amplitude and body bends were similar to wild type. In fact, they were a little bit more than in wild type. So what we saw was that compared to wild type, capture mutants actually showed a little bit more body bend and amplitude, which I've shown you previously, shows less. However, what we saw was when you made a double mutation between DOP2 and CAP2, you could completely suppress the DOP2 phenotype and the animals now look like wild. Now, if DOP2 is indeed affecting dopamine levels, and now you have no dopamine, you may imagine that it doesn't matter. If there's no dopamine, DOP2 has nothing to affect. So it will go back to looking like wild type. So this indicated to us that DOP2 is probably affecting dopamine levels, and it's not doing it by decreasing dopamine levels. What could DOP2 actually be doing? The next thought was maybe it is just increasing dopamine. If what you see with DOP2 is just increased dopamine levels, we thought, why don't we just treat wild type control animals with exogenous dopamine and see if they behave like DOP2 animals in the presence of ethanol. So this is wild type animals with ethanol, 2 hours of ethanol treatment, no dopamine. This is wild type animals with just dopamine treatment, no ethanol treatment. They both move around fine. Give wild type animals exogenous dopamine and put them on a plate with ethanol and image after 2 hours. You can see that they're hardly able to move. The anterior is trying to move and is trying to push or pull the posterior region along with it in a phenotype that is very similar to what we see with the DOP2. So we decided to quantitate this phenotype, and you will see that wild type with ethanol is the control animals. Wild type with dopamine looks very similar both in number of body bends and amplitude of body bends to just wild type with ethanol. Dopamine and ethanol shows a phenotype which is very similar to the DOP2 mutant, both in terms of number of body bends and amplitude of body bends. And you can see in both these cases, the posterior more effective than the anterior body. So the next question we decided to ask was, if DOP2 is indeed affecting dopamine levels, can we actually quantitate it? And to do this, we use a fluorescent tag, which is called fluorin. Now, what happens with fluorin is that fluorin fluoresces only when it's being released from vesicles. Okay, so when it's in the vesicles, it doesn't show fluorescence. Only when it's released, we see fluorescence. And how to do this experiment? We take a synapse where we have fluorin expressed it using synaptic promoter. So it's expressed in dopaminergic neurons using the SNP1 promoter. And then you bleach this area of fluorin and look at how quickly we get back fluorin. So now, if what we think is happening in DOP2 is that there's more dopamine, you will get back fluorescence in DOP2 mutants, which are treated with ethanol, faster than in control animals, which are not treated with ethanol, or wild type, or control animals, wild type animals, which are treated with ethanol. And that is exactly what we see. So, in the case of wild type, DOP2 without ethanol, or wild type without ethanol, the fluorescence recovery after the photobleaching experiment is very less, while in DOP2 mutants treated with ethanol, you get very quick fluorescence recovery after the photobleaching experiment. You can see here, this is quantitated here at 60 seconds and 120 seconds, that DOP2 mutants treated with ethanol show more recovery when compared to all the other control animals at both times. So this to us indicated that in dopaminergic neurons, when we remove DOP2, we likely have more dopamine free. So now the question was, okay, if this is the case, what neurons are actually involved in this process? I told you earlier on that we have 8 dopaminergic neurons. There are 6 of them in the head region and 1 in the posterior region. Now, just looking at the phenotype, we thought that maybe what is happening is that the posterior region neurons are the ones that are required for this phenotype. So we decided to posterior region neurons. And how we decided to do it was essentially decided to ablate this neuron from the animal. this phenotype is going through this neuron, then you can imagine that in DOP2 mutants, when you ablate this neuron, you will no longer get the phenotype because this neuron doesn't exist, so it doesn't matter if it's sending out more dopamine or not. That is a moot point. There is no neuron, it's not going to send out any dopamine. So you have the PDE neuron. If you were to remove the PDE neuron, irrespective of what happens is going to be, dopamine is not going to be sent out. This is experiment. So wild type, which we do a mock ablation phenotype. In wild type animals, when we ablate the PDE neuron, it looks just like the other wild type animals. All of this is looking at locomotion in the presence of ethanol. DOP2 mutants, mock ablation looks just like DOP2 mutants. They are unable to move in the presence of ethanol. The PDE neurons have these DOP2 mutants. They go back to looking just like wild type animals without or with the PDE neuron. This indicated that this phenotype is likely going through the PDE neuron, which accepts this state. So you can also look at the amplitude of body bands, and again, DOP2 mutants with mock ablation show a phenotype, while DOP2 mutants without the PDE neuron revert back to looking at wild type. So DOP2 appears to be functioning in the PDE neuron to allow for the increased dopamine phenotype. So in the PDE neuron, you remove DOP2, you're seeing increased dopamine. So the next question was, how does the dopaminergic neuron actually affect locomotion? Now, just to give you a brief idea of how neurons function through each other, you have sensory neurons, which are the neurons which are more external in the animal. These are what sense the environment. These neurons form synapses, neurons, which collate all the information together. The interneurons then synapse onto motor neurons, which synapse onto the body wall muscle and allow for normal locomotion. Is the sensory neuron. It synapses onto an interneuron called DBA, which synapses onto cholinergic motor neurons, which synapse onto the body wall muscle. So what we decided to do was we decided to look at the motor. We decided to look at the level of the motor neuron. This neuron is somehow affecting, not able to move properly. So obviously we affect the motor. We look at the motor neuron to see if this phenotype is going through the motor neuron. We were not using genetic trees for this. So we decided to use a drug. So what is known as that? It sends out the new color, and when acetylcholine is released, some of the acetylcholine is using acetylcholine. The rest of the acetylcholine binds the acetylcholine receptor and allows for normal activation. There are two possibilities here. Either what is happening is that there is acetylcholine at so in order to look for less acetylcholine, we looked at a large number of mutants which are required to process acetylcholine or to release acetylcholine, which is required for acetylcholine synthesis when you don't have the transporter that's required. So we looked at mutants of all of those and we found an artificial scenario where we have more acetylcholine. We did this drug. We use Avicap, which inhibits the activity of acetylcholinesterase. Now, if you inhibit acetylcholinesterase activity, what happens is that there is a large pool. Okay, so you're essentially acetylcholine at the synapse. The same experiment that we've done in the presence of life, in wild type work. How does this drug? So when you look all of them acetylcholine, this causes too much muscle contraction and paralysis in the body. So without the drug with just ethanol. And this is an animal why treated with the drug called aldecard and put on ethanol. And you can say that this active mutant on ethanol is trying to move very badly. The posterior is just not moving. It is trying to drag the posterior region of the worm along with it as so when we thought we should also use and we used an enzyme called which is a mutant in we used a enzyme called CHAT with choline acetyltransferase. So and choline are acted upon by choline acetyltransferase to give back to coenzyme A and acetylcholine. The CHAT enzyme, choline acetyltransferase enzyme, you should have very little acetylcholine. So even if you put aldecard, there's not enough acetylcholine to actually see acetylcholine acid in your muscle. So we use mutants as our control. These mutants are called CHA1. And you can see that compared to wild type mutants without aldecard in the presence of ethanol, wild type with aldecard are showing a phenotype similar to what we see in DOP2. When we use CHA1 mutants, they do not show much of a phenotype and similar to wild type without aldecard. This is CHA1 mutants which are treated with aldecard and then put on the list. And a similar thing was seen with body weight. You can see that one-time animals with aldecard treatment are the only ones that are showing a phenotype. So the next question was, does decreased level of acetylcholine cause DOP2 mutants to behave like wild type animals? Oh, sorry. I'm sorry. This is messed up. This. What we decided to do at this point was, okay, we also decided to use a second control. Take CHA1 mutants, put them along with DOP2 mutants. CHA1 in the presence of DOP2. Now, if DOP2 is actually going through this, and in the presence of DOP2, what we are actually saying is increased acetylcholine. So, CHA1, you have that downstream part is not there. You have less acetylcholine right away. So, the DOP2 phenotype, and this is exactly what this aldecard just with ethanol. Wild type shows no phenotype. DOP2 mutants show very obvious phenotype. Show phenotype, and when we make double mutants with CHA1 and DOP2, again we see no phenotype. DOP2, which shows increase in DOP2, is acetylcholine as its downstream part. It is actually causing more acetylcholine, and that is what is causing the worm to show the phenotype in the presence of ethanol. These are body against DOP2 mutants show phenotype. CHA1 mutant phenotype and CHA1 double mutants is just like wild. So appears to be due to insect at the neuromuscular level. So you have the PDE neuron, you have DOP2 in the PDE, DOP2, you have more dopamine, and this causes increase in acetylcholine at the neuromuscular junction. So the last question I wanted to ask was, what is the circuitry? How is DOP2 actually functioning in this PDE neuron to allow for changes in the acetylcholine motor? So when we went back to the literature, we found that the PDE neuron makes lots of synapses, 61 synapses. Okay, so C. elegans have approximately 7,000 synapses. 61 synapses between two neurons, that is almost 1% of all the neurons, which is a large number of synapses. So DBA neuron is sitting right at the tail of the worm and has a process that goes right to the head of it. With some of it which I had done my postdoc, showed that upon muscle contraction, calcium enters into the DBA neuron through a channel protein. This causes release of a neuropeptide, which is called NLP12 from the DBA neuron. And then binds to a receptor which is present on the acetylcholine motor neuron, and this causes acetylcholine. And this process keeps continuing and allowing for maintaining the correct amount of acetylcholine as a neuromuscular. Followed a few years later by work from Raja Bhattacharya, where he showed that the PDE neuron, which is dopaminergic, sends out dopamine that binds to DOP1 receptor that sits on the DVA. And that is also something that allows for this process to happen. So PDE sends out dopamine binds to DOP1 receptor. This then allows for NLP-12 release from the DVA. In the absence of DOP2, one would think that there is more dopamine binds to DOP1. This will cause more activity of DVA, which will send out more NLP-12. More NLP-12 will bind to the receptor on the acetylcholine will be released from the acetylcholine motion. So the question we decided was this actually functioning through DOP1 in DVA? And to address that, what we decided to do was we decided to remove both DOP1 and DOP2. Now, if you remove DOP1, then DOP2 will have nowhere to go. The more dopamine will have no, so you should say no phenotype. And then if we put back DOP1 just in this one neuron, in this one DVA neuron, can you then rescue this? So if you have too much dopamine, but you have no DOP1, here, so DVA will not cause more NLP-12. So what we looked at DOP2 mutants in the presence of ethanol, show phenotype, shown you all through this talk. On the other hand, do not show much of a phenotype. It shows slight phenotype, but you would not able to see it on the page at all. DOP1 and DOP2 double mutants behave just like DOP1 mutants. While if we want just into this one DVA neuron, we two ones which look just like the DOP2. So, and this was seen both as well as the amplitude points. Here, DOP1 again shows a bit. DOP1, DOP2 looks just like DOP1 in this DOP1, DOP2 double mutant, just one single neuron, just like the DOP2. The increase in dopamine from PDE in DOP2 appears to function through DOP1 receptors in the PDE. Now, the next question was, okay, if this is indeed the circuitry, if we were to just put more NLP-12, if we aim the DVA, could we see exactly the same phenotype that we see in DOP2 mutants? And that is exactly what we saw. We made 3 independent lines where we overexpressed NLP-12 just in the DVA neuron. In one of those 3 lines, we saw that they behave exactly like the DOP2 mutant. Ethanol, where they showed a phenotype where you have less number of posterior body bends and less number of amplitude bends and compared to that. They look just like the DOP2. You will not be able to differentiate. So finally, the last question we thought was, okay, if this is again just the circuitry, if we were to put a lot of acetylcholine receptor on the muscle, will we again type just like the DOP2? So we put a lot of acetylcholine receptor onto the muscle and we see a phenotype. It's not as strong as the DOP2 mutants, but it is a strong phenotype. The animal is not able to use normally in the presence of ethanol. This is DOP2 mutants. This is acetylcholine receptor overexpression in the muscle. We see a phenotype that appears to be similar to what is seen in the These are models for how DOP2 functions. We have DOP2 that sits in the PDE neuron in the posterior of the animal. In the absence of DOP2, more dopamine is released, which binds DOP1 receptor on the DVA neuron, which causes more NLP-12 released from the DVA neuron, which in turn binds to the CKR2 receptor on the acetylcholine motor neuron and causes more acetylcholine. And this is what, in the presence of ethanol, causes the worm to have defects in locomotion in the posterior of the animal, more so than anterior. It appears to actually be a fairly conserved mechanism in that when you increase acetylcholine, previous work has shown that in Drosophila, mutants that show increased acetylcholine synapses are more sensitive to ethanol. So these mutants are called arousal mutants. In Drosophila, they show increased acetylcholine synapses, And the incapacitating synapses correlates with the worm actually being more sedated in the presence of, with the flies being more sedated in the presence of ethanol in compared to control. Published some years ago, but we thought that this correlated nicely. With this, I want to end thanking the people who've done the work. This is the work by Pratima and Anuradha in IISER Mohali. The strains were from CBC and AdGene. Randy Blakely, Ron Evans, and Garcia lab for plasmids with Harjot Kaur and Anindya Ghoshal for the ablation experiment where we ablated the PDE neuron. And I want to thank E17.1 Mohali, SERB and IISc for funds, and thank you all for your time. This is my current plan in IISc. With that, I want to end, and I'm happy to take questions.
Yeah, thank you so much, ma'am. Yeah, thank you so much, ma'am, for a lecture on drinking and dopamine. What's the connection? Your presentation and witness that you have taken all of us to the new worlds of neurosciences and nematodes aspects in neuroscience. Thank you so much. It proves that your knowledge revolutionize in the field of neuroscience. Ma'am, today we had a large number of enthusiastic participants throughout your talk, and we have some general questions out of your lecture today. And I hope we can go through general questions, which are very general, that belongs to your experiences throughout your career. Shall we proceed with those questions?
Yes. Can you just wait for like 3 seconds and I'll be back. 3 seconds. I'm just going to have a drink of water. Is that okay?
Yeah, sure, ma'am.
Yeah, sure. Let's get started.
Yeah, sure, ma'am. Ma'am, can you elaborate your present work that goes on in a laboratory using your nematode? Habitus laboratory.
Yes. So some of the things that I'm interested in is I alluded to this early on in my talk. We're interested in understanding how cell surface molecules neuromuscular junction. So a lot of work so far indicates that cell adhesion molecules are required to bring the pre and the postsynaptic partner together. It's also known that they are required to maintain normal function of the synapse, normal structure of the synapse, but exactly how do they allow for release of neurotransmitters at the presynaptic site, or are they even involved in this process? Are they required to maintain postsynaptic receptor levels at the postsynaptic muscle or in postsynaptic neurons? So, and what are the signals? Who are they signaling to? So we found that some of them signals with the Wnt signaling pathway, because it's a very generic pathway that many of you must have heard about. So look at how these molecules, we actually get into very molecular, into the depth of the molecular mechanisms of who their interacting partners are, how they might be functioning. So those are the kind of questions that we are asking with respect to the neuromuscular junction. Apart from that, we're also interested in looking at neuropeptides, and I talked a little bit about neuropeptides here, but we're also generally looking at how neuropeptides are involved in locomotion. So we remove neuropeptides and see if they affect locomotion. That's what we're studying right now.
Yeah. Ma'am, why may I know why you are very restricted to this particular nematode or to this particular species?
One of the reasons is that it's very easy to work with. So if you were to look at mice or rodents or zebrafish, they're quite expensive to set up a lab with that. Nematodes, on the other hand, it's not cheap, but it's not that expensive. We can very easily look at these animals, and our interests are largely looking at it from a molecular perspective and to do a lot of genetics and mechanisms. Other than say C. elegans and Drosophila, are you able to do genetics? A lot of genetics that very, very in-depth genetics. These are essentially the two organisms if you want to look at the nervous system. I'm very interested. I'm a geneticist by training. I'm very interested in genetics. I, prefer to look at something where I can actually look very closely at what molecules are.
Still, before joining IISc, you have been associate professor in IISER Mohali. Still, you are a lion here in IISc. How you feel this research environment when compared to IISER and IISc?
I think they're very comparable. I think I really have not found that much of a difference. I think the environment is very comparable. Students are very comparable. My research is very similar. It's not very different in both the places. So I don't think that there's much difference.
Yeah, it's fine. I think
we have lots more trees, I think, in IISc, but it's nicer to
feel something similar, right?
Yeah, I'm actually from Bangalore, so IISc is more home to me. I've lived in Bangalore for many, many years before I left, so that's why I like Bangalore a lot. But otherwise, I don't think there's much difference.
Okay, ma'am, you obtained your PhD from National University of Singapore. Is this your PhD work lies in the same neurosciences background?
No, my PhD work was more in developmental and cell biology. Again, I worked on Drosophila, and we were looking at how the cytoskeleton affects protein localization, molecular, very in-depth genetics and molecular biology, but not the kind.
Yeah, yes, but you are in neuropeptides, neurotransmitters. What makes you to tense your interest towards neurosciences?
See, my interest is not that much in neuroscience; it's more in genetics or neurogenetics. I'm not a trained neuroscientist, or I like being able to make new looking at what happens when you lose a molecule, who the molecule interacts with. Just so happens that I'm working in neuroscience, but I mean, I don't think it's necessary that it's neuroscience. I think I'm more of a geneticist than a neuroscience. I don't think I'm a good person to answer this question.
Yeah, fine. As of now, you are so interested in neurosciences, right?
Yes, I mean, I'm interested in what molecules do at the neuron. So, what it is. It's not. I'm not interested in large questions of how do we think, consciousness. Those are not the kind of questions I'm asking. I'm essentially asking very molecular questions. And it just so happens that I'm asking this from a neuronal perspective.
Yes, sir. Then let's have some few questions from our participants, ma'am.
Sure.
Let me read out the questions. Sometimes cricketers score six in quick succession, 4 by 6 in an over. Can we believe it to have a dopamine effect in such cases?
Probably dopamine. I mean, definitely dopamine is involved because dopamine is required for normal coordination. So yes, but I think it's not just dopamine. I mean, I think adrenaline probably plays a good role in this case as well. neurotransmitters. I don't think you can just say it's because of dopamine. I mean, definitely dopamine is involved because locomotion, normal movement requires dopamine, but I don't think it's dopamine, just a dopamine.
Yeah. The next question is that how do we decide which organisms to choose for studying the neurological pathways and from which kingdoms?
So I think that depends on your interest. There are a lot of different organisms you can use to study neuroscience. So, like initially, Eric Kendall had used Aplysia to study aspects of neuroscience. People use Drosophila, zebrafish, mice, primates. I think it should. What are the questions that you're going to ask? Unless you actually know the questions you're going to ask, you won't know what's a good organism. So, if you want to see how eye movement happens, for instance, you definitely cannot use biology. They don't even have an eye. Probably Drosophila is not a good system because it doesn't have a comparable eye to humans. Primates may be a better system, or rodents may be a better system. I think I think the bottom line is it depends on the question you want to ask.
Yeah, sure. The next question is that how is artificial intelligence enhancing the research in neurology?
I think there's a lot of work being done in artificial intelligence. I think and in other sciences, you see a lot of artificial intelligence that is in play. I don't know much about it, so I don't want to say things that might or might not be completely safe.
Okay, ma'am. Yeah, fine. Ma'am, what are the opportunities and how international and national students can join your laboratory under your guidance?
So for PhD, I think there's IISc has a PhD program where students who scored something in some of these national exams are called for interviews. The process happens through interviews. So I think it's a very standard find out the details. Labs don't take students on their own. Students join the program, join the lab to take consultation. Both the PI and the student should be interested in taking in having. So yeah, I think that you'll be able to fairly easily find that online. Postdocs usually apply to the PI student join. So yeah, is there anything else I'm missing in this?
Yeah, it's all right, ma'am. Stop the mass.
They have an undergraduate program. I know I think you can go online and look to see what exams are required for that and how you get into that.
Yeah, sure, ma'am, sure.
I don't know.
Okay, ma'am, fine. Right. Yeah. Shall you drop a take on since our participants are from undergraduates, postgraduates, and the postdoctoral fellows, even faculties from different aspects or different streams of life? I want to see take-home message for them in this lecture by or like on this neurology.
So I think the take-home message is that basic fundamental research can be a lot of fun. You should pursue what you want to do, your passion. Start to work on what you want to work on. I think one interesting question. Most systems would probably turn out to be interesting. So you should do what you find interesting at the end of the day.
Yeah. Thank you so much, ma'am, for your time and patience, listening and answering all your questions. The speakers elaborate explanations. Thank you so much, participants. I thank our speakers of today's talk, Dr. Kavita Babu, for spending your valuable time with us. I extend my sincere gratitude to you on behalf of my entire team.
Thank you very much. Thank you. Bye-bye.
Thank you. Once again, I thank all for people who joined with us for today's talk. Thank you so much. Thank you, everyone.